The complement system is a crucial part of your immune defense, made up of proteins in your blood that work together to fight infections. It can be activated in three main ways: the classical pathway, the alternative pathway, and the lectin pathway. Each pathway is triggered by different signals, but all lead to a similar response, helping your body recognize and eliminate harmful invaders like bacteria and viruses. The complement system is activated through three distinct pathways that all lead to the same final destructive outcomes of opsonization, inflammation, and cell lysis. All three pathways converge on the cleavage of the central complement protein, C3, into its active fragments, C3a and C3b. 1. The classical pathway. This pathway is primarily activated by the adaptive immune system and is the link between the innate and adaptive immune systems. It is primarily activated when an antibody, IgM or IgG, binds to a pathogen surface, forming an antigen-antibody complex. This binding triggers the activation of a protein complex called C1, which then cleaves C4 and C2 to form the C3 convertase, C4b2a. This C3 convertase then cleaves C3. This cleavage splits C3 into two fragments, C3b and C3a-C3b. This is the larger, highly active fragment. C3b is crucial because it can covalently bind to the pathogen surface. This process is called opsonization, effectively tagging the microbe for destruction by phagocytic cells like macrophages. C3b is also the key component for the next step, as it can bind to the existing C3 convertase to form the C5 convertase. C3a. This is the smaller fragment. It acts as an anaphylatoxin, a chemical messenger that triggers inflammation by recruiting other immune cells to the site of infection and increasing vascular permeability. Summary: antibodies bind antigen. C1 complex activated and binds to the antibody. Cascade activates C2 and C4 to form C3 convertase. C3 convertase splits C3 to fragments, C3a and C3b, C3a responsible for inflammation and C3b responsible for opsonization. 2. Lectin pathway. This pathway is activated by mannose-binding lectin, MBL, or phycolins binding to specific sugar patterns on the surface of pathogens. MBL is an acute phase protein produced by the liver in response to inflammation. When MBL binds to mannose residues on a pathogen surface, it activates MBL-associated serine proteases, MASPs, which are similar to C1R and C1S of the classical pathway. Activated MASPs then cleave C4 and C2, where C4 is split into C4a and C4b, and C2 is split into C2a and C2b. The C4b and C2a fragments then combine on the pathogen surface to form the C3 convertase, C4b2a. This is the same C3 convertase formed in the classical pathway, which is why the lectin and classical pathways are sometimes referred to as the same from this point forward. C3 convertase splits C3 to fragments, C3a and C3b. C3a responsible for inflammation and C3b responsible for opsonization. 3. Alternative pathway. This pathway is activated directly by microbial surfaces, polysaccharides or other foreign materials. It does not require antibodies. The alternative pathway is initiated when C3 spontaneously hydrolyzes to C3, H2O. Factor B then binds to C3, H2O, and this complex is cleaved by factor D to form C3, H2O, BB, a fluid phase C3 convertase. C3 convertase splits C3 to fragments, C3a and C3b. C3a responsible for inflammation and C3b responsible for opsonization. This C3 convertase cleaves more C3, amplifying the pathway. Properdine stabilizes the C3 convertase on microbial surfaces, preventing it from being degraded. Some C3b molecules join the C3 convertase complex. This acts as the C5 convertase. Function cleaves C5 to C5a, which is potent inflammatory mediator, and C5b, which starts membrane attack complex. C5b binds C6, C7, C8, and multiple C9 molecules. This forms the membrane attack complex by forming pores on the pathogen, thereby leading to lysis. Functions of the complement system. 1. Opsonization. This is the process of coating or marking pathogens with molecules that enhance their phagocytosis by immune cells, such as macrophages and neutrophils. C3b is a major opsonin produced by the complement system. 2. Lysis. The complement system can directly kill pathogens through the formation of the membrane attack complex, MAC. 3. Inflammation. The complement system generates several anaphylate toxins, including C3a, C5a, and C4a, which are potent mediators of inflammation. 4. Immune complex clearance. The complement system plays a crucial role in the clearance of immune complexes from the circulation. Immune complexes are formed when antibodies bind to antigens. 5. Enhancement of adaptive immunity. While primarily considered part of the innate immune system, the complement system also influences adaptive immune responses. Complement activation products, such as C3a and C5a, can act as adjuvants, enhancing the activation of T cells and B cells. Thank you for watching. Subscribe.